you all for inviting me here and present some of the work we've been doing in this project. And most of all, thank you for funding this crazy idea. I don't think anybody else in the world will fund it until we actually show some convincing data. So I'm going to start by actually telling you the motivation for this project. And the motivation is based on breast cancer, and breast cancer is the main topic in my lab. And the reason because we're interested in breast cancer is for really two main points. One is that the social relevance of breast cancer and the economic impact that this disease has in the population. So basically, just to give you some brief statistics, a woman is diagnosed with breast cancer every 3 minutes, and it's every 20 seconds in the world. A woman dies of breast cancer every 12 minutes in the United States, every 60 seconds in the world. And roughly 1/4 million people is actually going to, 1/4 of a million women is going to develop breast cancer sometime this year in the United States. And 50,000 of them are going to die of breast cancer. When breast cancer becomes metastatic, it's actually very difficult to treat. There are no really good treatments for metastatic breast cancer, and these women, they actually die. Now, if you don't want to look at all the social relevance and you want to look at economic impact, look at the numbers. $5 billion per year is the cost of this disease in the economy, and this refers to having something like 80% of the people that is diagnosed with breast cancer being part of the population that is full-time workers, part-time workers, or self-employees. This causes a huge burden on the financial economy of each of the patients. Each treatment, for example, the standard treatment with Herceptin, for example, costs something like $80,000 just one round, and you need multiple rounds of Herceptin in order to actually undergo the therapy successfully. So that leads to people returning to work when they are not ready to go back to work because of the financial pressure. Some of them, they lose the job or accept jobs with different type of salary. So, but there are good news about breast cancer, and the good news is that if you detect breast cancer early on, 90% of the people is cured. So all we need to do is go and get our mammograms done and stop finding excuses for not doing it. And also, actually, 98% of the women that actually they have ductal carcinoma are cured these days. Now, if you have a metastatic process, meaning some lymph nodes taken, then that percentage decreases. So now, which one is the bad news about breast cancer? Well, the bad news is relapse. So 30% of the population actually relapses to breast cancer, okay? And relapse comes for women that actually have some, maybe a form of cancer called triple negative. Triple negative breast cancer is a very aggressive form of cancer, has very poor diagnosis. And basically, women diagnosed with this cancer, they receive a standard treatment, and that standard treatment, we know that it doesn't work. Relapse occurs between 2 years post-treatment and any time in the lifetime of the patient. That depends on age, genetic susceptibility, stage of cancer, treatment, lifetime, everything is bad, really, to be honest. So how is that this relapse happens? If you look at carefully to a process of cancer, basically what you have is a mutant cell that actually accumulates mutations over the lifetime. Every time that it divides, accumulates more and more mutations, keeps dividing until eventually it pops up and forms a tumor. And this tumor actually releases what is called tumor angiogenic factors, promotes angiogenesis, recruits blood vessels, you're going to see it here in a second, and these blood vessels actually bring oxygen and nutrients to the tumor, so the tumor keeps growing. If you dissect this tumor and look at what happened in the center of the tumor, what you're going to find out is the center of the tumor is necrotic. So most of the cells are dead. But the problem is not with most of the cells. The problem is with the cells that are not dead and they are in the center of the tumor. Those cells, they actually survive conditions as hypoxia, low oxygen. Actually, they're very resistant to very many drugs, resistant to radiation. They have very high rate of mutation. They stay dormant for very many years, and they don't differentiate. So why is this important? Because it's like having a little bomb inside your body. Those guys are inside your body, they haven't been removed, and one day, with no any reason, okay, that you can pinpoint, they're going to wake up and you're going to have a relapse in cancer, okay? So which one is our goal? Our goal is to get rid of those cells. So we want to literally fry them, okay? So this idea actually came, and this is the overall goal of the project. This idea came after listening very many of you talking, okay? So as a biologist, you can imagine that we don't have the in-depth knowledge of nanoparticles that you all have, but we know exactly what we want in the nanoparticles. So I kind of came up with this idea that is crazy, but I would like to have a superparamagnetic particle that actually is able to generate some kind of thermic, so photothermal therapy can be used and directed magnetically. It can be coated with another particle that actually contains ceramide, which is a lipid that actually is targeted on the mitochondrial membrane and disrupt the mitochondrial potential. And this particle will be targeted to breast cancer cells through what is called liposome pegylated that is modified with the folic acid on the surface. So what all that means is basically what I'm going is to generate a particle that is going to go to the cancer cell, it's going to get inside, it's going to selectively release ceramide, which is a lipid that is going to go to the mitochondria. We will be able to heat up these particles using a magnetic field, and at the same time, because the ceramide goes to the mitochondria, it's going to disrupt the mitochondrial potential. There won't be ATP, and at the same time you're heating the cell, you disrupt the ATP, the cell dies because it doesn't have any energy to survive. Make sense? It's like punch 1, 2 to the cell. Good? So it's like, well, I don't know. That's the way that I see it. So I came with this idea, and I said, well, I don't really know how to make these particles, but I have a lot of good people around campus that knows how to do the things. So I contacted him and I said, well, I need some kind of lipid where you can put in a particle and form a micelle, and this micelle can be selectively delivered to the cell. So he has this awesome system where he can generate this block dimer, okay, that forms a micelle. And this system is really nice because the chain ends is something that you can modify. So we can put a folic acid at the end, so we can generate a micelle that has this core 4-vinyl-imidazole group right here that has the advantage of changing the protonation state when it changes the pH. So if this guy goes inside the cell, goes to the endosome, basically what it does is gets protonated, releases the ceramide that is incorporated in the micelle, and at the same time destroys the endosome because of osmotic lysis. So basically, his group was able to generate something that is pretty close to what I need. We just need to incorporate the folate now that is stable, and it has been stable for at least 24 hours. And our idea is to actually put this in cells and actually monitor the translocation of ceramide to the mitochondria, which I don't think will be any problem. So at the same time that he was doing all this work, we were working in the lab with the next phase, which is, okay, let's assume that the particle is done and we can put a folate there, which I don't think is any problem. So how are we going to make sure that the particle is uptaken? How are we going to make sure that it goes to the endosome, that the ceramide goes to the mitochondria and induces apoptosis? So we did a really big number of experiments. First, we have in my lab a collection of 63 cell lines, breast cancer cell lines that comes from primary tissue. So these are cell lines that actually are totally related to a tumor and behaves like the primary tumor. We catalog them with the source, clinical, and pathological features. We map over 700 genes in the cells, so we know exactly how they behave at the genomic level. We monitor endocytosis mediated by folate, using a folate-pegylated liposome that is synthetic that we just bought. Calculate the number of receptors on the cancer cells. We study mitochondrial localization, and this is a really nice localization using different approaches. We study the release of the production of ATP, the respiration on cancer cells, and the metabolism in these cancer cells. And we also monitor how the apoptotic cell death is happening in these cancer cells. Why do we need all this stuff? Because we want to be ready by the time that the folate is incorporating those particles. So now you're going to look at me and say, Carla, this is really cool, but you're trying to solve a 3D problem using a 2D system, because these guys are still in tissue culture. So that was bugging me for a while. So what I did is I recruit this kid, which is my student Michael, and we built a 3D printer in my lab. So we changed the pipette for screwdrivers, okay? But don't tell the biology people. So we actually built a 3D printer, and this came all from Nicole's student. That's the meeting that I attend once for your student and Pablo's student. So they use a matrix with collagen to grow cells, and I thought it was a fantastic idea. So we built a 3D printer where we can create a scaffold of collagen with vessels inside and grow cells that they are actually different in number and heterogeneity. And we can put cells that are resistant to drugs in the middle of a tumor and see how the cells behave in a 3D system when we treat with the particles that we are designing. So goals, really the more important things for now is to get 1 or 2 pops with my collaborators to be sure that when we submit the proposals, we are believable as a team. Okay, we already submitted the DoD grant, got feedback back. The 2 feedbacks were get a medical school associated and be less ambitious. And so I'm working on that part, on the medical school part. Work on sending grant to the triple negative breast cancer foundations. My goal is an NIH-R01. We have enough preliminary data for an NIH-R01 in June, and we already submit a couple of pre-submission inquiries to some agencies. So that's pretty much it. Sorry.
Thanks, Carl. Great. Deanna, I think you're up next.
So Tiana's going to be talking with us about nanomorph theranostic devices for controlled delivery of neuropharmaceuticals to the brain.
Okay, I want to thank Ictus and Bio for funding this project at the stage when it was back with the envelope idea. And so I'm going to show you the progress that we've made. So really we got inspired by this figure here about so-called next generation personalized medicine drug delivery system. And if you look at this figure and this cycle here, ideally you want to have a system that you can load with any kind of drug that